Advanced snakes and heliodemated lizards are the only two lineages that are known to have evolved venom delivery systems. Research has shown that there are 9 toxin types in which are shared between these lizards and snakes. While they have different abilities in which the venom works, the toxins appear to be fairly similar. Although it was found that the iguanian lizard Pagona barbata had a different venom gland secretory system compared to the advanced snakes and the anguillemorph lizards, there is found to be overwhelming evidence supporting that there was a single early origin of venom systems in these reptiles. The research began by identifying that the anguillemorphs, iguanians, and the snakes are the only lineages that have protein-secreting mandibular and/or maxillary glands. This was done by comparing the different families of reptiles and finding whether they have these venom glands or not. Although they all have evolved completely different venom glands, they all have this in common, that they have the protein-secreting venom glands. In this figure, all these reptiles have a relation to each other. Although we are only focusing on the venom clade, it can be noted that all these reptiles have a common similarity. It can be seen in the purple that the serpents have maxillary glands in which they store their venom. Although they both have maxillary glands and the mandibular, they use the maxillary glands for the venom compared to the Helmintidae, the Anguidae, and the Verandae, which have the mandibular glands, which is in the orange. The Iguania uses both mandibular and the maxillary glands for their venom storage and secretion, which can be seen in the red. These are the differences that can be seen between the venom clade. One experiment was conducted to show the effect of the venom's toxins on a rat's blood pressure. This was done by giving intravenous injections of Varanus varius to anesthetized rats, demonstrated a rapid drop in blood pressure. This showed that natriuretic peptide action of relaxation of the aortic smooth muscle. The graph that can be seen shows how the blood pressure dropped dramatically as soon as the venom was administered to the rat. The blood pressure dropped from around 90 millimeters of mercury to around 40 millimeters of mercury in under 10 minutes, and then began to rise slowly after this time. By the analysis that has been done, it can be reasonably assumed that these reptiles did come from a common ancestor. Although it cannot be fully determined who the ancestor was or where it originated, it can be seen in the venom clade that they have common similarities in the venom and the way the venom is stored. So therefore it is reasonable to assume that this could be a hypothesis that should be considered when comparing these reptiles.